In this video, I'll talk about blood clotting, the logic of blood clotting, why does it work the way it does, how it happens, and some of the trade-offs in the amount of clotting. To fully understand this video, you need to understand protein-protein binding, release of chemical messages, ligand-receptor binding, second messenger systems, activation of microfilament assembly proteins to assemble microfilaments inside cells, enzyme activation by cleaving away a portion of the enzyme or phosphorylation, and enzyme function. What starts a clot? Some injury severs a vessel here or cuts into it. That triggers a release of chemicals from the damaged cells that bind to receptors on smooth muscle of the vessel wall. Those receptors then activate second messenger pathways that allow calcium into the cell and cause contraction of the muscles nearby. And so what was a wide opening becomes a narrow opening. And I won't sketch that because that's part of what I'm expecting you should know before going through this. Then what are the events inside the blood vessel? So those signal molecules from damaged cells, they are binding to receptors on platelets. So I'm going to start sketching. Here is a platelet. It's a fragment of a cell. If we zoom in, here is a platelet. Here's another one. They break off from cells called megakaryocytes and wind up as little membrane-bound non-cells, but with cytoplasm inside. And they change when there is damage. They change in response to signal molecules from being these smooth round to being these spiky structured objects. So to sketch those, what we'd want is to show a platelet with, here's some chemical message coming from damaged cells, binding to a receptor, activating a second messenger pathway that then activates the assembly of microfilaments. So that second messenger pathway, if we blow this up, here's a much bigger example here, our second messenger winds up activating a protein that starts assembling microfilaments nearby. So here's an active microfilament assembly protein, and so actins are now being assembled to form these spikes. I'm not sure that it operates in this way. This is the most logical way I can think of to make it happen, but there are some other possibilities. And so this second messenger then causes all these spikes with a core of actin within each one. What's the advantage of those spikes? Well, if you've got a broken end of a tubule, these little round things are really easy to lose. But if we do two things, one is if we change those from being round and smooth to being spiky, and if we shrink this even a bit, that will help. Now you've got these things are likely to start tangling up with each other. The spikes are going to interact. They also become sticky. They have surface proteins that will stick to each other, binding in some way to each other, so that you wind up with this mesh of spiky little structures that are made from the platelets that stick to each other and stick to the wall of the vessel with surface proteins that are binding to each other in some way. So where the smooth ones would have gone through, these spiky ones do not. Now, at the same time as this is going on, the other part of clotting is the formation of fibrin strands, which you can see faintly, I hope, in this image. Here is a fibrin strand, here's another one, here's a fibrin strand, here's a fibrin strand right there. And those fibrin strands start to make a mesh. So here's some of this mesh helping to trap these spiky platelets. And between them, they start stopping the flow of blood. Red blood cells get trapped against them. And so we would add to this a mesh of fibrin. And the fibrin is going to attach at the edges to extracellular matrix proteins. The extracellular matrix proteins appear to be mainly one called fibronectin, that is a major part of extracellular matrix and of the basement membrane, which is a specialized extracellular matrix that is around the inner layer of cells in a blood vessel. And so that helps to anchor the fibrin mesh. So let's talk about how we get that fibrin mesh, but I hope so far you've got the idea of some chemical message binds to a platelet, causes these spikes, and activates a protein on the surface in some way to make it more sticky, to make it bind to other proteins and to on other platelets and to the walls of the blood vessel. And then what about that fibrin mesh? Well, the fibrin mesh is made by this clotting cascade. Critical points are that we have a fibrin, which is what is forming this cross-linked fibrin clot that's trapping platelets and red blood cells, like in a fishnet. Now, you don't want active fibrin all the time, or you'd have clots forming all the time. So the fibrin is held as an inactive cell, an inactive protein, fibrinogen, in the blood. It is a major blood protein. You have a lot of it. It's one of your most abundant ones, and it's made mainly by cells in the liver that produce this fibrinogen. And free fibrinogen does not stick to itself. They don't stick to each other. As fibrin, they start forming these complex meshes. So you need some way to activate it. The simplest way to get a clotting cascade going would be to have an enzyme that will do the conversion. You actually need to cleave some of fibrinogen off to make fibrin. And the simplest way would be to have injury would cause something to make prothrombin, an enzyme, convert to thrombin, an active form of that enzyme that catalyzes this conversion, cleaves off, in this case, part of the fibrinogen to make fibrin. So injury causes prothrombin to turn into thrombin to make fibrin, and you make clots. However, instead, we have some other enzyme, factor 10, interacting with factor 5A, that converts prothrombin to thrombin. So injury must somehow activate this event, and that happens with yet another pair. In this case, there's two ways to do it: 9A active and 7A active. These are blood factors. They were just discovered in the blood as factors, nobody knew what they were originally, that affected whether or not clotting worked effectively. And so they just got names of factors 6, 9, 10, whatever. So we have one step here. We've got another step here. In order to get these active 9A or 7A, we've got yet another step up here. Two ways to do it. Either 3 will do this one, or 11 will do this one, converting inactive 9 to active 9. And up here we have yet another step, and starting with damage to the vessel wall. So why so many steps, and why an intrinsic and an extrinsic pathway? There's some logic to this in that there's two areas in which you might want clotting to happen, and the speed and the amplification might differ. An important thing to realize with all of these is that each of these enzymatic steps involves an amplification. So if we have damage to the vessel wall, which starts this pathway, a part of it is actually activation of platelets to release some of their contents, which starts this pathway. Then we might have the damage to the vessel wall causes release of some molecules, and that release of molecules at the first step might activate one of inactive factor 12 here to active factor 12A. At the next step, maybe each factor 12A activates at least 10 factor 11s. So at the next step, we've gone from 1 to 10. We've got an amplification factor of 10. And at the next step, maybe 11A can activate 10 9As. And so we go from 10 now to 100 active enzymes. And at the next step, we maybe do that again. We've gone to now 1,000 active 10As. And each 10A can activate 10 thrombins. We now are at 10,000. And the activation might actually be even greater than that. So we've gone from one original molecule. Each original molecule that binds to a target might activate 10,000 thrombins. And then, of course, each of those thrombins can over and over again activate more and more fibrins. So the reason for the length and complexity of this pathway, one major factor, is simply amplification. If this is actually what's needed, we might expect to see some variation across animals that's related to how many amplification steps they need. Animals with high blood pressures, high pressures in the circulatory system, like mammals, like us, we have this full pathway. And we need that pathway because with our high pressures, we can bleed to death incredibly quickly. And so anything we can do to get quick clotting tends to be favored by selection. So this quick process to get a lot of fibrins in a hurry is has been favored by selection. These amplification steps mean that the first few bits of damage start this whole clotting pathway, so that very quickly you've got tens of thousands of thrombins activating hundreds and thousands or millions of fibrins. However, animals with lower pressure circulatory systems don't have the same selection pressures to clot quickly. And in fact, there's a trade-off here. Faster clotting with fewer molecules released to trigger it has a risk of unwanted clots. And that is a serious problem for humans. We can get bruising or other sorts of damage that causes clotting that we don't want, and then the clotting itself may be dangerous and even lethal. Whereas animals with low pressure circulatory systems, they can afford to have fewer steps, and in fact that is what you see. It's only animals with the highest pressure circulatory systems that seem to have both the amount of fibrinogen that we have and the sheer number of amplification steps we have. So with a lower pressure system, you can have fewer amplification steps and lower risk of unwanted clots. Why have both an intrinsic and an extrinsic pathway? And might there be selection pressures that explain why the intrinsic pathway here has more amplification steps than the extrinsic pathway? The intrinsic pathway has 2 amplification steps more than the extrinsic pathway. The answer might lie in the fact that this is a damaged vessel wall. And in a damaged vessel wall, you would want to repair that as quickly as possible, especially given that a lot of the fluid may be moving rapidly away from the area of damage because of the pressure of the blood behind it. And so additional amplification steps may help compensate for the fact that a lot of these chemical messages are being carried away. That result then would be that you'd get clotting that is starting faster in that location to help repair the damage. Now, the extrinsic pathway happens because of trauma damage to cells outside of the circulatory system. So imagine a cut that both cuts a vessel wall and damages cells nearby. That is due to the interaction between these clotting factors and extracellular proteins or messengers. And there, it may not be quite so critical to have fast clotting, and so the selection may favor fewer amplification steps because that reduces the risk of excess unwanted clots, and it also reduces the cost. The more of these proteins you make, the more costs, and the more ways there are for it to go wrong. Finally, the clots, the fibrin doesn't just stick to itself. The fibrin does self-assemble. It has binding regions that make it self-assemble into these actually very orderly structures, and that the fibrin connects especially to, it appears, fibronectin. Fibronectin can be bound to, so fibronectin is an extracellular protein. It can be bound to integrins that are bound to actin microfilaments on the inside. And fibronectin is bound to extracellular matrix components like collagens or elastins or glycoproteins. But fibronectin also occurs in other locations than just attached to the integrins, and it appears as though fibrins can attach to fibronectins. And so you get this mesh that then is tethered to nearby extracellular matrix. The result would be a blood vessel at a cut in which you'd have this mesh of fibrin trapping red blood cells and trapping spiky little platelets and tethered to extracellular matrix nearby. There are some further steps. The fibrins get covalently linked to each other by another enzyme once they've formed into this mass, and then the whole thing shrinks some and tightens up to give a strong plug.